Hello everyone, in this video we're going to be talking about how to analyse DNA using Gel Electrophoresis and a process also known as southern blotting. Now this is really aimed at Key Stage 4, Key Stage 5, and I'm not going to go into great detail in terms of the genetics of how we extract DNA, for example, or how we digest the DNA in this process. I'm merely just going to outline the steps involved in gel electrophoresis, how it works, the principles behind it, and why you would choose to do it, for example. So let's start with the why. Why you would need to do this. This process allows you to run what are called lanes of DNA samples. And you can look at the DNA samples and it allows you to fragment that DNA, to put it into different sized fragments, to be able to analyse connections between two different DNA samples. So you could, if you're looking, for example, at a sample of DNA from a crime scene and you want to compare it against a standard or a known, or any other known sample, you could do this technique and look for similarities in terms of the DNA fragment size. Equally, in paternity testing, this kind of DNA test, if you like, allows you to look at similarities between a potential father and their offspring. You look for similarities in the fragment size, which would give an indication of similarities in the base sequences of the samples. That's why you might want to do this. But let's talk about the process and the steps involved. So the first thing you would need to do is what we call extraction. So that's the first stage. So what we need to have ultimately in some kind of, we'll just draw a very basic test tube here, in some kind of test tube would be DNA extracted from a sample. Now, if that sample is too small, we could use techniques like the PCR, polymerase chain reaction, to increase quantities of it, and I've talked about PCR and its method in another of these videos. We all have repetitive core sequences of DNA that are actually non-coding, called introns. Now, those aren't really much use to us. And those core sequences differ for different people in terms of sort of their size and the number that we have. But that's just something as a side thing to note, that we do have repetitive core sequences as part of this DNA. Once we have extracted the DNA that we need to then undergo a process of digestion. So what we're going to do is ultimately, if we just draw it in red, we're going to want to use what's called the restriction endonuclease to cut that DNA into fragments, actually at specific recognition sites. Again, I'm not going to go into recognition sites in this video, but we need to digest that fragment, or that sample of DNA rather, into fragments. That's the second thing we're going to do. After digestion, that's where it becomes a little bit more involved, because we're going to need a technique of separation. So for separation, the fragments need to be separated as such using what we call gel electrophoresis. So we have a sort of plate. If I try and draw this as best as three-dimensional as possible. So we have this plate with different lanes running, and I'll just colour this in here. And you get this kind of pattern forming. So we have here what's called a gel plate, and this, if I will show the direction of movement, so movement is from A to B here. Now what we have at the top, at A, would be our large fragments, and at B we have small fragments. Now this is how electrophoresis works. Electric probes are attached to the gel plate, a positive terminal one end and a negative terminal the other. Now DNA is negatively charged, so we can imagine that A would be our negative end and at B would be our positive end. So those negatively charged DNA fragments that have been digested up will move clearly towards the positive end. They're attracted to the positive end. Now a question that my students ask is, why do you think the fragments move and how does size affect them? Well, they move because they're attracted, but the fragment size is important because if you're a smaller fragment, you'll move further and faster. So that's why I've put that large fragments are found at A and smaller fragments are found at B, because the smaller fragments move quicker and further, so they'll move further down the gel plate in the direction shown from A to B. So once we've separated out the fragments, we then have a fourth stage, and this is also called separation, but it's separation part 2, if you like, because the DNA fragments are transferred from that gel to a nylon membrane in a process known as southern blotting. So I'll draw this out again. I mean, it's not the best drawing by any means, but it ultimately just shows the principle I'm trying to get across here. So we have our plate with our lanes. I mean, clearly there'd be a more distinct banding pattern in the real sample, but I'm just drawing this for the sake of argument. And what you do is lay on top of that a nylon membrane. So this here is a nylon membrane, and that goes on top of this gel plate, and that's called southern blotting, because by putting the nylon membrane over the gel plate, those DNA fragments get transferred to that nylon membrane. So once the DNA fragments are transferred to the nylon membrane to roughly the same positions where they were in the gel, we expose it to then UV light. So once we've put the nylon membrane into the gel plate, or we've picked up those DNA fragments, we expose that whole nylon membrane to UV light. And what that does is just to help fix those fragments and keep them in position. So that's what we do in separation part 2. Now I'm going to shrink this down a little bit to continue the process underneath. So we've had separation. Now what we need to do is stage 5. And stage 5 is called hybridization. Let's think about what happens in hybridization. So we've had this nylon membrane that's been exposed to UV light. Now what we're going to do is add DNA probes to label the fragments. These radioactive probes attach to specific fragments via complementary base pairing. So the whole purpose of hybridization is to attach radioactive or radiolabeled DNA probes. So basically, if you take that nylon membrane laid in a tray, so if we just draw, for example, here's our tray, and at the bottom of the tray we have our nylon membrane there with our strands. What we're going to do is basically just pour into that a sample of radiolabeled DNA probes, and that's called hybridization. Then they'll attach to specific fragments via complementary base pairing. Now this happens under very specific conditions, and you need a very high temperature for this to occur. So once we've had hybridization, all that's required now in our final stage is development. And that's our final stage in this gel electrophoresis southern blotting technique. We'll take our nylon sheet with radiolabeled DNA strands and that membrane, if you like, with our radiolabeled DNA is placed onto an X-ray film. So we have our membrane here, a nylon sheet with our radioactively labeled DNA, and we place that onto an X-ray film. Not the best diagram again, but it's just more for a visual to show the steps in this process. So here's our X-ray film. We place it, a nylon sheet onto it. Now, development of the X-ray film would reveal dark bands where the radioactive DNA probes have attached. So what you'll ultimately end up with is, and I'll show this at the top. What you'll end up with would be something that looks a little bit like this, and you may have seen this on diagrams in books or on the internet. But you'll end up with a sheet with characteristic sort of bands that look a bit like this, and I'm just doing a rough sketch. This is no means accurate. But you end up with an X-ray film revealing dark bands, and that's where that radioactivity has been detected, where those DNA probes have attached. And all you have to do is look across the bands to see where these sort of fragments line up. So you can see, for example, we've got a similar banding pattern here and here. We've got a similar banding pattern here and here. You just look horizontally across and see where those banding patterns match. Now, in a separate video, I talk more about those banding patterns and something called Sanger sequencing and restriction mapping. But in this video, all we're talking about is how you analyze DNA in a process known as gel electrophoresis to form that banding pattern. Okay, I hope all of that helps.